So hi, hello and welcome again. Microbehunter here, I'm Oliver, and today in this video I want to give you a couple of tips on how you can collect water samples, like I've got over here, for microscopy, so that I can put it under the microscope to see what some of the microorganisms and some of the critters that are in here. I'd like to observe them under the microscope, and there are a couple of tips I would like to give you also because I made a couple of mistakes in the past myself. Yeah, so we're going to have a look. I have to tell you, I don't know exactly myself what's in here, so maybe it's going to be really cool, maybe it's going to be really disappointing. That's always a little bit one of the fascinating things about hobby microscopy is you don't always know exactly what you're going to see. But before I do that, I would like to give you a little bit of background and a little bit of context where I collected this water sample from. Of course, you can already see a video on the side here. And I spent the last weekend at the so-called at the Wörthersee, which is in Austria. It's about 6 hours by train from where I live. So I traveled across Austria just to this lake to collect this water sample. Well, of course not. No, I was there for personal reasons. One of my kids, he was participating as a chess competition, and every time when I travel with him, I take along a couple of those plastic tubes, and I like to basically collect samples that I then take home, and then I put them under the microscope, and that's basically where we are. So what I would like to do now first is I want to give you now the first tip when collecting water samples, and that's already the one that you're able to see here. You notice that actually I'm not just collecting water, but that I'm actually collecting some solid material. You are not going to find a lot of interesting things in the water itself. The concentration of microorganisms and algae and other things is actually too low. The water there is pretty clear and it's pretty clean. So you have to collect some solid material from the bottom. So I've been scratching off some of the stuff from rocks and some sediment, and that's actually the interesting thing here, right? And this is where you're able to find in the biofilm. That's the stuff that's growing on rocks and on the surfaces. That's full of life. Even right now, it's the beginning of March. The water was ice cold, really. My fingers were freezing off, but we still can expect something. So what I'm going to do now is, that's basically tip number one. Now let's have a closer look here. And let's put everything. So I've made a little. So here we are again. You can see a little rack here. Here's the water sample. And the second important thing, and I would say that this is really absolutely vital, and that's the biggest mistake I made, is never ever even think about filling up the whole tube to the very top. Don't do that. What's going to happen is then the sample is going to become anaerobic. This means there's not enough air, not enough oxygen present, and then it's going to start to smell like sulfur. So anaerobic bacteria are going to start decomposing it, and when you open it and smell it, it's going to smell like bad, right? This one here actually smells. Smells perfectly fine. The reason is because it's not anaerobic, so you can actually keep it with the cap on here without problems for several days actually. Usually at home, what I do is I remove the cap to allow for enough air exchange. But when then the water evaporates, I add a little bit of more water. So it does maybe pay off to take a little bit of extra water along as well. So that was tip number two. And the third tip you've already seen yourself, and that is the tube should be transparent. It should allow light to go through because there are of course algae in here, diatoms in here, cyanobacteria in here. They need light for photosynthesis, and they also produce oxygen for other microorganisms that are in here. So basically these are the three tips. Don't fill it up, collect solid material, and make sure that the tube is transparent. If you kind of wonder what tube this is these are so-called centrifugation tubes. You can see that this one over here has a conical bottom. That's a disadvantage, but there are a few of those as well which you can actually stand up. They come in various sizes. I have got a few others over here as well. There's something in here already. No, here that's empty. You can see over here there are also smaller ones. But generally, I like those. So let's actually do some practical observation. Of course, you need the sample. You need a slide. I've got a cover glass over here. And now the problem is the following. How are we going to get the sample out here? I use a pipette, of course. And the important thing is now the following. You need scissors. These are the pipette tips. Let me move this away here. And they're a little bit too thin here. So they're not able to get a lot of sample material out. So I usually take some scissors and I cut the tip off simply to make it a little I've got the pieces flying around my room now. So it's a little bit, as you can see here, a little bit wider. So I can also yeah, take up some solid material. This is a microliter pipette. This is how it looks like. Yeah, it's actually one of the cheapest ones I could get. They can be super expensive, really, but I do not need them to be very, very precise in any case, because I'm not going to be measuring volumes. I just needed to actually pick up some of the material here. And then you want to take, of course, some material from the very bottom, go all the way in, and hope to, yeah. Let's see if there's something in here. Okay, there's not a lot. Let's try another one. And here we go. Okay, and we put this back. I drop a cover glass on top. This one over here. And you see, it's not even completely flat, and that means that there is maybe some grains, some sand grains, or I don't know, or some algae or whatever that basically prevent the cover glass from completely going flat. It's going to be okay. So let's have a look. Okay. Yeah, you probably have already also noticed, if you've been watching my videos for a longer time, that I've changed the format around. Yeah, now you can see you can see now the stage a little bit larger, and I need to turn on the microscope, of course. Here we go, and let's have a look. No, you always start at the lowest magnification, of course. Yeah, so that's basically what we see over here. A little bit as expected, not a lot of movement going on here. We still have to wait a couple of weeks maybe until the temperature goes up. But if you think it's just a little bit messy, well, there are a few interesting things that are there. Yesterday, when I had a quick look, I could actually see a nauplius larva, which is the larva of a water crustacean, a little animal. But I think we have to go a little bit higher. Here we are now seeing a few more details. These are that's a whole diatom colony here. So I think that's a pretty huge one. But let's have a quickly scan the slide to see what we're able to find here. Anything that's a little bit greenish, of course, is photosynthetically active. Yeah. So over here, let's have a look at those. Here, this is a small diatom colony that we're able to see here. I think we have to go up a little bit more with the magnification. Yep. So diatoms are single-celled, and they are unique because they have a shell made of silica, which is glass, essentially, a cell wall. Okay, so they have a cell wall. You shouldn't say shell, because the term shell in biology means actually something different. It's made of, shells are made of calcium carbonate, and that's something that animals have. We call this a frustral. Hope I pronounced it correctly now. But let's have a look. As we can see, not so many. Not so much movement, but if we go up with the magnification, we are able to see a few diatoms moving around maybe, and I hope that I'm able to show you that. Yeah, see that this long thing here, that is an empty diatom. Yeah, you can even see that it's a little bit damaged and broken. This one here must be still alive because there's a green chloroplast on the inside. Yeah, am I not lucky? Look over here. I finally did find a gliding diatom over here, right in the center. No, it stopped. Apparently, scientists are not entirely sure how they move because they seem to be producing some kind of a slime layer along which they're gliding. Here we go again. Okay, yeah. So you see that there's plenty of life here in this small water sample. And again, you have to do a little bit of trial and error, and then maybe you're able to find something interesting. And then with an identification book, you can do a deep dive, and then try to identify what they actually are. Yeah. So. And I think from my side, other beautiful diatoms over here. Look, from my side, that's actually all I want to share with you today. I hope that you found this video informative. Again, I hope that with my 3 pieces of advice, you're also going to be successful finding some interesting microbes, microorganisms, and algae and whatnot under the microscope. All the best from my side. Happy microbe hunting as always, and see you in the next video. Bye-bye.